Okay, we're going to move on now to a very brief discussion of the urinary system. And the reason it will be a brief discussion is that much of what needs to be understood about the kidney is not really gross anatomy. The gross anatomy of the kidney is not all that important clinically or important for USMLE questions. What is important about the kidney, of course, is the renal physiology, blood pressure regulation, acid-base regulation, the histology. All of those are really quite important, but not the gross anatomy. There are a couple of issues about the kidney that I think have some gross anatomical relevance, and that's all that we'll talk about. If we think of the kidney as having two parts functionally, and as it'll turn out, these two functional parts correlate with two developmental portions of the kidney. The two functional parts of the kidney are what we would call the filtration portion of the kidney and the conducting portion of the kidney. The filtration portion of the kidney is the part of the kidney that filters blood and makes urine. In other words, the nephrons. And for the most part, not precisely, but for the most part, the filtration portion of the kidney is in the cortex of the kidney. It's mostly in the cortex of the kidney. The other part of the kidney is the conducting portion of the kidney. By that we mean the part of the kidney that takes the urine that's been produced by the filtration portion of the kidney and conducts it or moves it someplace else. So the conducting portion of the kidney, which is mostly, not entirely, mostly in the medulla of the kidney, is made up of the collecting tubules, which is what the urine that comes out of the distal convoluted tubules gets dumped into. So we have thousands of collecting tubules that you can see represented here by these streaks heading toward these renal papillae, which then empty into the minor calyces. And then the multiple minor calyces empty into a few major calyces. And then the major calyces empty into the renal pelvis. And then the renal pelvis is continuous with the ureter. So the urine that's been produced by the filtration portion of the kidney gets carried away from that part of the kidney through collecting tubules to minor calyces to major calyces to renal pelvis to ureter. The important anatomical feature to note here is that as that urine is moving in the direction I just mentioned, it's going from smaller to larger to larger to larger diameter structures, with the minor calyces larger than collecting tubules, major calyces larger than minor calyces, renal pelvis larger than major calyces. So that means that it's not possible for a renal stone, a renal calculus, to get impacted, get stuck as it goes from smaller to larger diameter structures. The first time that the renal calculus can get impacted is the first time that it goes from a larger diameter structure to a smaller diameter structure. And so the first time that happens is right here at the what's called the uretero-pelvic junction, the junction between the ureter and the renal pelvis. And so a stone, as it's attempting to leave the renal pelvis to get into the ureter, if that stone is larger than the upper end, than the diameter of the upper end of the ureter, it's not going to get through. It's going to get impacted there. However, if the stone is small enough to get through that opening, it will proceed down the ureter. And now let us take a look at the next slide. We see that the ureter, after leaving the kidney, proceeds down the posterior abdominal wall. And throughout its course in the posterior abdominal wall, it's sitting right on the anterior surface of the psoas major muscle on both sides. That's going to be an important landmark for us when we look at CT scans. And then the ureter crosses from the abdomen into the pelvis at the pelvic brim here, which coincides with the bifurcation of the common iliac artery. So it crosses right over the bifurcation of the common iliac arteries to head into the pelvis. So that's the pelvic brim. As the ureter does that, it constricts a little bit. So that means that a stone that was small enough to get through the uretero-pelvic junction likely will not get impacted until we get to the level of the pelvic brim, when the ureter narrows down. So it may get impacted there. So that's the second location. But if the stone is small enough to get past there, then it will continue on down the posterior wall of the pelvis, across the floor of the pelvis. And if I just jump forward for a moment, we can see the ureter at this point entering the back wall of the bladder. As it goes through the bladder wall, it narrows down again. That's called the ureterovesical junction. Ureterovesical junction. So that means a stone, let me back up, a stone that got past here and got past here still may get stuck as it attempts to enter the bladder. So since that is the smallest of the three passageways, that's the most likely place for a stone to get stuck. Even the smaller stones might get stuck there. So as you would anticipate, the most common site for a renal calculus to be impacted is at the ureterovesical junction, just as the ureter is going through the bladder wall. If the stone is small enough to make it through the ureterovesical junction, then it will not get stuck because the ureter getting into the bladder is much smaller than the urethra coming out of the bladder. So if the stone can get into the bladder, it can get out of the bladder through the urethra. So the three sites for renal stone impaction are at the ureteropelvic junction, where the ureter crosses the pelvic brim, and at the ureterovesical junction where it goes through the bladder, with the most common site being the ureterovesical junction. Okay, let's turn our attention now to the development of the kidney and ureter. Now, we had seen this illustration back at the beginning of our discussion when we first talked about the development of the gonad. And at that time, we pointed out the ridge of mesoderm found on the back wall of the abdomen. We called it the urogenital ridge, a ridge of mesoderm on the back wall of the abdomen. And we said that most of that urogenital ridge will be related to development of kidney structures or urinary structures, and a smaller portion will be devoted to gonadal development. And at that time, we talked about the gonad portion. So now let's turn our attention to the rest of it, the part relating to the development of kidneys. So on this slide, which is illustrating development of the urinary system, we see a different image representing that urogenital ridge. It's now begun to differentiate. So this is that same very long urogenital ridge. And we see different parts of it are being given different names. The most cranial end of it is being called the pronephros. The large middle portion is being called the mesonephros. And the most caudal end is being called the metanephros, or metanephrogenic mass. So this ridge of mesoderm is going to differentiate into urinary structures, into filtration structures. But it does so in an interesting way. It doesn't all differentiate at the same time. There is a wave of differentiation that goes from a cranial to caudal direction. So this differentiates first, and then this, and then this, and then this, and so on down the line. So there's this wave of differentiation. So the part that differentiates first is called the pronephros. The part that differentiates later will be mesonephros, and that differentiates in a wave. And then lastly will be metanephros. But also interestingly, following behind that wave of differentiation, there is a wave of degeneration. So while the mesonephros is differentiating, the pronephros is degenerating. And when the more caudal mesonephros is differentiating, the more cranial mesonephros is degenerating. So we have a wave of degeneration following behind this wave of differentiation. So this doesn't all exist. Pronephros, mesonephros, metanephros does not all exist simultaneously. They occur sequentially. The pronephros, the very earliest differentiating structure, differentiates and degenerates very, very quickly, very early in development, and in fact never functions as a urine-producing structure in humans. So the pronephros is really just vestigial. It persists as a structure in lower species, but not in humans. so we don't have to deal with the pronephros. The mesonephros, on the other hand, does in fact, in the early embryo, does in fact filter blood and produce urine. So the mesonephros is the early functioning kidney. Now, since it has tubules that's producing urine, that urine needs to have a place to go. And so what we see running right along the side of the mesonephros is a duct called the mesonephric duct. So the mesonephric duct runs along the side of the mesonephros, and the urine that's produced in the mesonephros gets dumped into the mesonephric duct. The mesonephric duct carries it down, and if we follow the mesonephric duct, we see it empties into the very caudal end of the gut tube. Here's the gut tube, and here's the caudal end of the gut tube. And you see that the caudal end of the gut tube is enlarged, is dilated. And that caudal end of the gut tube is called the cloaca. Cloaca means sewer. So, appropriately enough, the urine is getting dumped into the sewer end of the gut tube. There is a membrane separating the cloaca from the amniotic cavity, but pretty early in development that membrane ruptures, so the cloaca is now open into the amniotic cavity, and so that means urine that's being produced can be dumped into the amniotic cavity. And in fact, that's what happens throughout fetal life, embryonic and fetal life. Even later in development, when it's no longer the mesonephros, but the definitive kidney that's producing urine, the urine is being dumped into the amniotic cavity, and most amniotic fluid is in fact urine, fetal or embryonic urine. That explains why, when there is agenesis or any other developmental defect that interferes with either urine production or urine conduction, so that urine is not being dumped into the amniotic cavity in its normal rate and volume, there's going to be less than the normal amount of amniotic fluid, and that's called oligohydramnios, too little or abnormally little amniotic fluid. And so oligohydramnios is going to be an indicator of urinary system defect, either an obstructive defect or renal agenesis or something of that sort. Before we finish our discussion of the kidney, let me just finish that thought about amniotic fluid and urine. Since urine is continuously being dumped into the amniotic cavity, we'd expect that urine has to go someplace, because the amniotic cavity is not just going to get larger and larger and larger. And in fact, what happens is that the fetus swallows the urine and then reprocesses it, so that we have this continuous production of urine being dumped into the amniotic cavity and swallowing of the amniotic fluid. So we have basically a dynamic equilibrium of amniotic fluid. But that means that if there is an abnormality that interferes with swallowing, that we're going to end up with too much amniotic fluid. And that defect that interferes with swallowing could be either a GI tract defect, an obstructive defect, let's say atresia of some portion, let's say duodenal atresia as an example, or esophageal atresia as an example, where it interferes with the ability to transport amniotic fluid that's being swallowed, so you'll get a buildup of amniotic fluid. Or if the GI tract is perfectly intact, but the neurological connections, the neurological control of the swallowing reflexes is defective because of central nervous system defect, such as anencephaly, then that fetus, again, will not be swallowing normally. So in either case, you'll have a buildup of amniotic fluid, and that's called polyhydramnios. So oligohydramnios is an indicator of renal tract defect, and polyhydramnios is an indicator of either GI tract or neurological defect. Getting back to our discussion of the developing kidney, we know that this mesonephros is going to be degenerating, and it will eventually be replaced by what will become the definitive kidney down here at the caudal end of that ridge, what's listed here as the metanephrogenic mass, or the metanephros. So in fact, after the mesonephros is no longer producing urine, there is no need for the mesonephric duct, and it is lost in females. There's no longer a need for it, and it degenerates. However, in males, even though the mesonephros is no longer producing urine, and therefore there's no need for the mesonephric duct as a urinary conduit, remember that the gonad, the testis, is developing right alongside of the mesonephros, and in the male, the gonad does need a duct, the ductus deferens. So in males, the mesonephric duct, after it has finished serving its urinary function, gets converted over, and it is what becomes the ductus deferens. Okay, so the mesonephric duct in males is retained as the ductus deferens. In females, it degenerates. Now, let's turn our attention to what will become the definitive kidney, which is going to be arising in this caudal end of this ridge. And if we look at the next slide, we see an enlargement a little bit later in development. Here's where that mesonephros was that we were looking at before, and here's at the caudal end. This is that metanephric blastema or metanephrogenic mass, all means the same thing. And here's that mesonephric duct. And we see the mesonephric duct entering the cloaca as we said it was going to. Let me call your attention to this little branch right here that comes off the mesonephric duct. It grows out of the mesonephric duct. The name of that branch is the ureteric bud. The ureteric bud. So it's a branch of the mesonephric duct, and it grows from the mesonephric duct toward the metanephric blastema, or the metanephrogenic mass. Now, we said that this metanephric mass or blastema is going to become the definitive kidney, but that's not precisely correct. What is correct is that this is what's going to become the filtration portion of the kidney. In other words, this will become the nephrons, largely in the cortex of the kidney. But importantly, that mass of mesoderm that's being called the metanephric blastema here will not differentiate into nephrons unless the ureteric bud grows to it and induces it to do so. So the ureteric bud, in addition to what it's going to become, which we'll talk about in a few moments, in addition to what it's going to become, has a very important inductive relationship in that the ureteric bud induces the metanephric blastema to become the filtration portion of the kidney, to become nephrons. Then, interestingly enough, after this metanephric blastema begins differentiating into nephrons, it in turn has an inductive relationship back onto the ureteric bud. And it causes the ureteric bud to start to branch, to dilate at its distal end and then to branch. So the dilated end of the ureteric bud is what becomes the renal pelvis. The initial branches from the renal pelvis, from the ureteric bud, will become the major calyces. The branches of the major calyces will become the minor calyces, and the branches of the minor calyces will become the collecting tubules. So the ureteric bud not only becomes the ureter, it also becomes the conducting portion of the kidney, largely the medulla of the kidney. So the ureteric bud becomes the ureter, the renal pelvis, the major and minor calyces, and the collecting tubules of the kidney, while the metanephric blastema becomes the nephrons of the kidney. So each of those structures has an inductive relationship with the other. First, the ureteric bud induces the metanephric blastema, and then the metanephric blastema in turn induces the ureteric bud to start to branch. The ureteric bud, we can see, we said it's a branch of the mesonephros. We've already said that the mesonephros drains into the cloaca. All of this, as it enlarges, will get incorporated, so we'll end up with the ureteric bud eventually getting incorporated into the wall of the cloaca. And so we have the ureter entering the distal end of the gut tube. Now, that distal end of the gut tube, the cloaca, is going to end up being subdivided by a septum, a mesodermal septum called the urorectal septum, which grows down and takes this cloaca and divides it into a front portion and a back portion. The front portion of the cloaca is going to become the urogenital sinus. The back portion of the cloaca is going to become the anorectal canal. The urogenital sinus will become, in part, the urinary bladder. So now we see that the ureters are entering the cloaca, the part of the cloaca that becomes the urogenital sinus. The urogenital sinus becomes the bladder, so we have the ureters going into the bladder, which is what we want to have. Putting together two thoughts that we've already expressed, one that the mesonephric duct in males becomes the ductus deferens, and the second that the ureteric bud is a branch of the mesonephric duct, and the ureteric bud that induces the metanephric blastema to become the kidney, that explains why when in a male, if you find a male who's missing a ductus deferens on one side, you can be pretty sure that that male will be missing a kidney on the same side, because the absence of the ductus deferens implies that there was an absence of the mesonephric duct. If there was no mesonephric duct, there would be no ureteric bud. If there's no ureteric bud, the metanephric blastema would not be induced into forming a kidney. So absence of the ductus deferens implies absence of the kidney. And finally, we see that coming out of the urogenital sinus, there is another duct that goes out into the umbilical cord. This is a vestigial duct. It has no real function in humans. It's called the allantoic duct or the allantois. In humans, it usually becomes fibrous and disappears very early in development. However, if it doesn't disappear, if it doesn't become fibrous, you're going to be left with an open communication from the urinary bladder out into the umbilical cord. That's going to be called a patent urachus. The allantois or allantoic duct, when it normally becomes occluded, changes its name to urachus. If it doesn't become occluded, it's called a patent urachus. And so if you have a patent urachus, when the umbilical cord is severed, you'll have created an open communication between the outside world at the umbilicus and the urinary bladder, and you'll have urine leaking out at the level of the umbilicus. That would be indicative of a urachal fistula, a failure of the allantoic duct to fuse. Of course, you can have partial fusion where most of it fuses, but a little region doesn't fuse, just like we've talked about in other developmental processes, and that would give you a urachal cyst. So that concludes our discussion of the development of the urinary system.